Tecnologia é o que move o mundo, mas poucas pessoas sabem o que as novas plataformas tecnológicas podem fazer pela nossa qualidade de vida e na prevenção de doenças. Aqui em Biomanguinhos trabalhamos todos os dias em busca de pesquisa, inovação e desenvolvimento tecnológico. Somos uma unidade da Fiocruz que atende prioritariamente as demandas da saúde pública nacional, desenvolvendo produtos imunobiológicos que fazem a diferença na vida de milhões de brasileiros. O trabalho de desenvolvimento tecnológico é dividido em 4 programas: vacinas virais, vacinas bacterianas, kits para diagnóstico e biofármacos. Esses programas contam com diversos laboratórios, núcleos organizados por plataformas tecnológicas, equipes altamente qualificadas em inovação incremental e transferência de tecnologia. Essa estrutura permite que os projetos aptos avancem à fase de estudos clínicos e produção em escala industrial. Durante a pandemia da Covid-19, nossos laboratórios atuaram na busca de soluções e outros produtos essenciais para o Sistema Único de Saúde. Desenvolvemos projetos de vacinas próprias que podem oferecer autossuficiência do país na produção do imunizante. Uma delas é a vacina sintética, com base em peptídeos capazes de induzir a produção de anticorpos para defender o organismo contra o vírus da Covid-19.
Essa vacina, ela identifica pequenos pedacinhos do vírus SARS-CoV-2 por bioinformática, e esses pedacinhos eles são acoplados em nanopartículas de ouro para desenvolver respostas imunológicas que combatam o vírus SARS-CoV.
Nossas equipes também trabalham no desenvolvimento da vacina de subunidade, que utiliza proteínas do vírus ou seus fragmentos capazes de estimular a melhor resposta imune.
As formulações vacinais testadas mostraram resultados muito satisfatórios na fase de imunogenicidade e toxicidade dos mesmos.
Outro projeto é a vacina de RNA autorreplicativo. Para sua produção, são escolhidos antígenos específicos do vírus para compor a formulação. São utilizadas diversas estratégias especiais para a obtenção das melhores respostas imunológicas para a proteção do organismo.
O RNA contendo proteínas do vírus SARS-CoV-2 é encapsulado em lipossomas que vão entregar esse RNA na célula do indivíduo vacinado. Dentro da célula, esse RNA ele sofre um processo de multiplicação e também dá origem às proteínas do vírus SARS-CoV.
Biomanguinhos está estruturado para atuar tanto no desenvolvimento, análise da eficácia ou estudos clínicos de protótipos de vacinas. Desenvolvemos kits para diagnóstico molecular e testes rápidos, inclusive os antígenos e anticorpos utilizados nos mesmos.
No segmento de diagnóstico, nós temos que destacar alguns resultados concretos que foi possível, então, oferecer e devolver para a sociedade. Nós trabalhamos no desenvolvimento e na implementação da produção escalonada do ensaio molecular para SARS-CoV. É uma ferramenta imprescindível desde o primeiro momento para o enfrentamento da pandemia, e nós conseguimos produzir 500 mil testes e oferecer isso por semana para as ações de saúde pública no Brasil. Com relação aos testes rápidos, é uma plataforma dominada tecnologicamente aqui em Biomanguinhos, na Fiocruz, e que nós temos a possibilidade de desenvolver e produzir inúmeros testes, inúmeros kits para diversas doenças. Mas agora a gente já está implementando e utilizando os testes de detecção de antígenos do SARS-CoV. Esse teste de detecção de antígeno, com base na plataforma de testes rápidos, permite que em até 20 minutos tenhamos uma efetiva resposta se aquele indivíduo tem ou não o SARS-CoV. E agora, mais recentemente, estamos em fase final de desenvolvimento para implementação e uso amplo dos testes de detecção de anticorpos neutralizantes para SARS-CoV. Isso significa que nós poderemos, de forma ampla no Brasil, ter uma avaliação das pessoas que possuem já uma efetiva resposta contra o SARS-CoV.
A pandemia trouxe grande interesse de diversos setores da economia na busca de soluções, não apenas no desenvolvimento de vacinas, mas também na automação de processos essenciais no combate à doença.
Biomanguinhos vem investindo muitos recursos em tecnologia, inclusive na automação de processos. A automação ela auxilia consideravelmente na rotina dos ensaios de sorologia, uma vez que ela aumenta a qualidade desses dados gerados, a capacidade de testagem e também a velocidade na liberação desses resultados.
Tais avanços significativos foram alcançados graças à estrutura e capacidade técnica de Biomanguinhos. Esses recursos e serviços também estão disponíveis para parceiros e instituições externas, visando o crescimento da área de biotecnologia do país. Competência, infraestrutura, inovação, pesquisa e tecnologia de ponta. O objetivo de Biomanguinhos é oferecer as melhores soluções nas áreas de prevenção, diagnóstico e tratamento de doenças. Trabalhamos todos os dias para cumprirmos o nosso compromisso de levarmos mais saúde a todos os brasileiros.
Boa tarde a todos. Good afternoon, everyone. First of all, I would like to thank you the presence of the audience here in the auditorium and also the people that are connected virtually with this panel. Today, we're gonna initiate the panel, the second panel of the International Symposium on Immunobiologicals, where we will discuss the strategies to accelerate biopharmaceutical development, driving innovation to speed up access. To comply with that, we are gonna have two lectures. One lecture will be given by Dawn Stewart, that is present here in person, and another lecture by the Thomas Gerlach. So, presenting the bio of Thomas Gerlach, he is a pharma professional with more than 30 years of experience in biopharmaceuticals and venture capital, who worked in big multinational companies like Novo and Novartis in Germany and Switzerland, but also in successful startups like Actelion in Switzerland and Brazil, early startup like Biozeus in Brazil, and in venture capital in life science. During his career, he worked in drug development, leading global teams, but also in commercial and operational functions as general manager of Actelion Brazil. During his 9 years in venture capital, he identified investments opportunities in biopharmaceuticals, diagnostics, and medical equipments, and supported the portfolio companies on the operational as well as board level. Currently, Thomas is Chief Strategic Officer of Penex Therapeutics, a New York-based biopharmaceutical company developing first-in-class Penex one-channel blockers targeting chronic pain and other indications. Thomas holds a PhD in biology and a bachelor in chemistry from the University of Mainz in Germany, and has spent 3 years as research at Michigan State University. Now present Dawn Stewart. Dawn Stewart is the CEO of Plant Pharma Corporation and is an entrepreneur and a scientist with 30 years experience in the biotechnology industry. Dr. Stewart is a founder of the Canadian biotechnology companies, Plant Pharma Corporations, and Antoxa Corporation. As the CEO of Plant Pharma, Dr. Stewart has advanced the company's plant-based manufacturing technology over 10 years from its startup, directing fundraising and development of a strong intellectual property portfolio and implementation of international partnerships with pharmaceutical companies, government organizations, and foundations, including Fiocruz and Biomanguinhos. Dr. Stewart gained the experience in biosimilars as the Director of Research and Development for Cangene Corporation, a biotechnology company in the Apotex organization. At Cangene, Dr. Stewart led in-house and intellectual collaborative programs developing biosimilars and innovative drugs. Dr. Stewart graduated with a PhD from the University of London in UK, and gained academic experience at the University of Alberta in Edmonton, Canada. So special thanks for Dr. Don and Thomas that we with us virtually for accepting the invite and contribute to our discussion with their experience, knowledge, and sharing their work. So Thomas, feel free to share your slides and you can initiate.
Okay, many thanks for the introduction and many thanks for inviting me. Actually, the slide should be uploaded from your side. Okay, now it's coming. Go please to the title slide, the next. Can you change please to this title slide? Okay, when I first saw the title of my presentation, I thought it's very broad, and I tried to, in my speech, my presentation, I tried to narrow it down a little bit more on focus on drug development for novel mode of actions, novel drugs with new mode of with new targets. And also I narrowed it down to the interaction in terms of outsourcing, the interaction with CMOs, CROs, and academic partners. Before I go into the, let's say, discussions on outsourcing development activities, next slide, I would like to remind you first, can you please switch the slide? Next slide, please. The slide before, please. To remind you what drives drug development, because this has an important impact on how you manage your interaction with service providing partners. It sounds very trivial, but the core mission of drug development is translating basic science into novel therapeutic products. And here I want to underline products. This translation from science into product is not that easy as it seems. Because once you develop you design your development product, your development plan, you have to be clear that you have a clear idea of what at the end is your target. What is your target product for profile? Because at the beginning, you might not know everything about your molecule, your mode of action, how it works, how the side effect profile is. But you have to have a good idea of where you want to be at the end, where to go. Because only this way you ensure that the development program supports also at the end the therapeutic use and the final product. And here you have to sit down sometimes and really look at your data, because we are in a data-driven environment where the data will drive the development path. And you have to look back on whether what you have done so far, the data you generated so far, support your target at the end. These are the famous decision milestones, because if this is not the case, you have to go back and you have to redefine your target, or totally or kill your product or project, or redefine totally your total development strategy. And also never forget that in our business, before you can launch your product on the market, there's another hurdle, and this is the regulatory approval. Therefore, your product development program has to be according to the standards of FDA and Anvisa. Otherwise, you will not reach the market. Therefore, we are talking about a long, risky, very expensive and complex process which has to be managed. It requires multidisciplinary collaboration and a product-oriented approach. And here we come more to the point, how to manage this in the most effective and also economic way. The critical decision points in drug development is always time, risk, quality, cost, and flexibility at the end. The next slide, please. Don't expect that your development path is straight. It's not. It's not just going from A to B. Many times you have to go back. The data do not support your path. You have to go back, rethink, redo, or there are barriers in the way. Therefore, it's a long and complicated way. And to manage this and the collaboration with potential partners is very complex. And you have to always keep in mind that a development path is not that straightforward, and you need the flexibility. Next slide. When it comes to executing your development plan, it's always a question, how much can you do in-house versus contracting out development activities to development partners? This could be an academic partner. This can be a CMO, a CRO. There are many potential partners which can help you to bring your product, your project to the market and transform it into a project, into a product. When I started my professional career in big pharma, the company, all the companies were fully integrated companies, and we did everything in-house. Today, also in big pharma, it's a mix of in-house and outsourced, which is today the standard. When it comes to startups, many times you don't have the choice. Many startups start as totally virtual companies. My own company, Panex Therapeutics, is today still a total virtual company, because we do not want to invest at this stage in lab space and the cost, the headcount to run an own lab. Therefore, once you can come to the question of outsourcing versus in-house, you have to always look at what are your core interests. What is your know-how? Do you want to have more flexibility? What is the potential of cost saving and risk sharing? And not to have to invest in your own lab infrastructure. And this is a trend which goes throughout our industry. Initially, most CROs and CMOs were based in the US and in Europe. But recently, in the last decade, or a little bit more even than this, there was a huge expansion to India and China, making the choice of CROs more and more complex. But it's a model which is deemed more effective from both sides, from the pharma as well as the CRO perspective, because it has a benefit that it gives you flexibility, sometimes reduces time, reduces bureaucracy, and can optimize your development program. But there is a big, big but. You have to do it the right way, because otherwise it can generate delays and a lot of problems. Next slide. If you want to outsource, there are two questions you have to ask you. The first and one important, what should be outsourced? Because that's not so trivial. Not everything makes sense. If it's a virtual startup, you don't have the question, but if you have in-house capacity, you have to think twice what to outsource and what not to outsource. I will come back to this a little bit later on. The second important point you have to ask, which is the right partner? Because the selection of the right partner will define the success of your partnership with the service provider. And the third point, and probably most important point, how to manage the company CRO relationship successfully. To be clear, most of the time, it's not the technical part, the competence part of the CRO order on your own side, which really makes a project fail. Most of the time, it's communication, lack of common understanding, of simply what is important and how to combine both organizations in a way that they work synergistically and you get the maximum benefit out of it. And this is not that simple. The question of what should be outsourced. The decision drivers in the business are usually always the same. It's competence, time, risk, cost, quality, and flexibility. If you have, for example, standard, let's look in the scenario of a CRO, standard studies, standard talks, standard ADMA studies. These studies are available from CROs off the shelf. They have routine, they do this routinely. They can do this relatively fast and probably more cost efficient than you can do it. Therefore, there is no question that it makes a lot of sense, and big pharma today sources out everything which is standard and is not their core competence to partner companies. Specialized models, when you come to specialized animal models and studies, things get a little bit more complicated. If it's outside your core competence, you can develop it on your own, but you can also look for a competent partner which has already some experience in it. And this could be an academic partner, it could be a commercial partner, and a CRO. And you contract this organization to develop the model for you and establish it and conduct the study. Many times, this is the better solution. Once you have capacity restrictions, tight timelines, outsourcing is always a good option to overcome these restrictions. And the cost argument, cost reduction, speed, are always important decision points for the decision whether to outsource or not. At the end, it's always a mix of what makes more sense for the company. And this drives at the end your decision on what to outsource. When it comes to the right partner, it's getting a little bit more complicated. You can, again, look into competence, time, geography, cost, quality, flexibility. But many times it's also a question of big versus small CROs, or a big versus small partner. There are these Wuxis in China, or big US CROs. It has advantages and disadvantages to go for big partner. If it comes to standard programs, sometimes to go with a big partner makes a lot of sense, because you give in one hand a lot of studies. You don't have the hassle of shipping compound from one to the other. You have to deal only with one interface and one team on the other hand, or other side, which decreases your management efforts to manage these programs. But when it comes to very specific needs, specific indication, specific studies and experiments, many times small CROs are the better choice, because they have specific know-how and are very specialized in certain therapeutic areas or certain models or certain technologies. Therefore, you have to see how the scientific capabilities of the CRO or your partner, again, whether it's an academic or a commercial partner, to meet your needs. But you have to spend some time in really to figure out what makes most sense for you. The geography is another consideration you have to always keep in mind. different time zones, different language. Sometimes cost is not the only driver, because many times Indian CROs, also they have today already reached a good level, and also Chinese CROs are a good option in terms of cost-wise, and they can provide good service, but geography is sometimes a barrier, and you have to keep this in mind. And again, you have to balance both sides. Communication, if you already, when you negotiate and select a CRO, when you have already not a good feeling on how they respond to you, how they communicate, how easy it is to get the right person, and the response time is slow, then it's already a bad sign, and you cannot expect that this will get better over time. Therefore, in this case, you really think twice whether you go in the partnership with this CRO or this university. Timing, cost benefit, quality are always critical points. And don't forget at the end, the acceptance by regulatory authorities. Does this CRO or this partner have an FDA history? Because at the end, the acceptance of the data produced by you and your partner plays a role in the approval process. Therefore, as a summary, when there is no match from the beginning on, the success of this partnership is already jeopardized. Therefore, spend some time here to really find the right partner, because it's difficult. Once you start it, there's no way back, or you will lose a lot of time. The third point is, in my opinion, the most critical point: how to manage the company CRO relationship successfully. Here, it depends a lot on your side and the other side. If it comes to CROs, the first point, as a clear contractual relationships, is usually no point, no issue, and also a discussed and signed and agreed study protocol. When it comes to universities, this is already a major barrier. Never start a relationship and in a partnership with a university, with an academic institution, if you don't have an agreed study protocol and signed a study protocol. When it comes to once you have initiated your project, it's key you have accountability on your side. You have to define someone from your team who is accountable, who is responsible for this project, and the same on the other side. Who is in charge? This is essential that you over time you have a good relationship and fast progress of your project with this CRO or this partner. Effective communication lines. Communication is all. You have to develop a solid relationship with your partner. You have to have agreed timelines and how you follow up. This has to work very efficiently and take here into account the cultural differences and sometimes language barriers. I can tell you Indian English and Chinese English is sometimes a challenge, and therefore be prepared for this, and also take into account sometimes the cultural difference, because sometimes it depends on the cultural background. The answers you get, they are not always that straightforward as you might expect, and therefore take into account that in some cultures they are not so straightforward, and then you have to really insist a little bit and and see that you're always on the right path with them. Assign a good project manager. And with good, I mean, it's not just someone who does time tracking and activity tracking. You need someone who sees the big picture, who's able to sense and to see when something fundamentally goes wrong, and when the management has to be alerted, and that this could jeopardize the progress and success of the project. Therefore, it has to be someone who really, again, sees the big picture and has a good overview of the project and where you want to go. A total alignment of your outsourcer, I mean, your CRO, and they have to understand also to a certain extent your processes, your people, and some to a certain level also a certain level of insight into your project. Because as more they understand what are your needs, where do you want to go, as more they can adapt and to better to your needs. And last point, treat the outsourced team, I mean the team from the CRO side, like your partner, as you would expect it to them, vice versa. Yeah. Therefore, good product management and activity tracking is here key. Here, from the business side, you always look at time, quality, and cost. But to get to your project delivery or to your target, but there are a lot of other points. It's very complex, and you have to always consider a good governance, the satisfaction changes which can happen. We are in a data-driven environment. The risks, we always have to be aware of the risk and have to always be aligned on whether the risk we are taking today is still adequate in order to reach our final product target. Therefore, it's just to round this a little bit up. The last slide. It's in summary, there are 3 pillars for success in outsourcing. First, be clear. You have a clear strategy and you have to see the clear benefit of what you want to outsource. Second, who is the right partner? CRO, academic institution, CMO. It's a mix of points which will decide at the end what is the right partner for you. Expertise, size, quality, geography, whatever. And the most important point, project management and communication. You have to manage this well from both sides. Only this will make this to a success. Okay, thank you very much. When you have questions, please let me know later on in the discussion section.
Thank you very much, Dr. Thomas. It was excellent, very clear. I think it's pretty important to us to see how to manage and complex projects, outsourcing and doing virtual PAD. Sometimes we can't see that in real, but we see that it happens and it's attendance. Now we are gonna start another lecture with Dr. Don Stewart, please. And he will speak about plant-based production platform as a strategy for acceleration of the development of biologics.
Okay, hi. It's a great pleasure to actually be here in Rio today. Not just because we're meeting our colleagues who we're working on a complex project with, a drug development project, but because the temperature in Rio is 32 and it was -1 in Toronto at my house this morning. So we're very happy about that. So we're a Canadian company. Small company, 20 people. And in the context of the discussion here, we're very pleased to be in a partnership with Biomangenis developing a drug. And Biomangenis bringing a lot of expertise, know-how, and capability in analytical and manufacturing to using our technology. So I'm going to really focus on why we would use plant-based technology for manufacturing a therapeutic. But let me tell you a little bit about our company. We're based in the Toronto area in Canada. We have a subsidiary company called Antoxa that's focused on the countermeasures to bioterrorism. And we also have a wholly owned subsidiary company here in Rio, and we're in a partnership with a local company in Rio. We have two areas of business, the biodefense area in Antoxa and the biosimilar business, both developing products which have a shortened time through the clinical path to market, so reduce cost, reduce time, and providing a good opportunity for a company to develop. The key candidates we have in the biodefense side are an antidote to ricin that was developed by the Department of Defense Canada. There's been a partnership with the Four Eyes group, that is the US, Canada, Australia, and Canada, UK, developing this, and we've now licensed the product about 2 years away from market. We have an antidote to sarin gas. On the biosimilar side, we have an biosimilar Lucentis, wet macular degeneration drug, and the project we're working with Biomangenis on is a biosimilar Keytruda for market entry on patent expiry in 2028. So when we're looking at biosimilars, we're trying to find the most cost-effective way of production. And, oops, I've gone too far. There's a variety of different strategies being used by different companies to produce biosimilars. And in many cases, companies have used the same type of technology. So either bacterial systems for non-glycosylated proteins or mammalian cell systems for glycosylated proteins. But not every case the originator technology is used, because if you developed a drug 20, 30 years ago and it's coming off patent, the whole way of making mammalian or bacterial fermentation has changed. And so the modern methods, which are most cost effective, are not necessarily the best. But here we have a variety of different approaches that have been used to produce oh, sorry, I'm talking about the wrong slide. These are, sorry, I got ahead of myself. Anyway, there are a variety of different systems used to produce biosimilar drugs. Now, what we're looking at here is plant-based expression. We are a plant-based expression company, and there's a variety of companies working in this area. Medicago in Canada just got approved by Health Canada to produce for a COVID vaccine. It was just a couple of weeks ago. Protalix, who you would know here, generated Licio, biosimilar drug that's approved and was marketed by Pfizer, and a variety of other candidates. So when we're looking at alternative manufacturing systems, and particularly plants as the manufacturing system, there are pros and cons. Plants are very low cost, and I'll talk to you a little bit more about that. The facilities are low cost and the timelines are low cost. But there are disadvantages, and as a developer and in our partnership, we have to be cognizant of these things and concentrate on mitigating the risk. It's interesting if you look at the number of approvals of recombinant pharmaceuticals produced in different systems. So this is a projection that was published in Nature from coli derived in purple to the orange one, which is mammalian cell derived. These are biosimilars, actually. And then there's the anticipated plant produced materials, I see both biosimilars and innovative, and then other types of plant-based sequencing. So you can see that in around about 2020, from then on, plants have become much more accepted by the pharmaceutical industry and by regulatory agencies. So So what is this technology? So we use a type of plant called Nicotiana benthamiana. It's a type of small tobacco. And this is some growing a biosimilar Herceptin molecule. It's a fairly simple process. You grow the plants for 4 weeks in a fully contained environment. At the end of 4 weeks, you've grown up an agrobacterium, a bacteria that only infects plants, that contains the gene of interest and some of the mechanisms of control. And that's what's called infiltrated. It's forced into the plants, and the plants are grown for another week. The genes jump from the agrobacterium onto the plant chromosome, and the plant starts producing your product. So after only a week at this point, you can harvest, extract a drug, and then you purify the drug with standard ways. So the main thing that we're looking at here is timelines. And if you start at the bottom, which is the take-home message, we can determine if we can produce a drug in 14 days once you've got the gene sequence. This is compared to months and months, years in most other systems as well. And to go scale up to grams, we're talking days as opposed to months or years. It's a huge advantage. When we're looking at expression development, we can not only have these short timelines, but we can actually overlap things so that we can look at different iterations. We can look at different vector systems, different control, different media on a week-by-week basis, very quickly coming to a conclusion as to the best way of advancing a product. Another aspect of this technology is the ease of scale up. Mammalian fermentation is complex, can take years to scale up, whereas with plants you have identical plants that are grown in an identical controlled environment, and to increase their production, you just increase the number of plants growing. It's very simple. This is an example on the left of a vertical farming system. It's called a gravity flow system, 13 layers high. It's 15 meters long by 3 meters wide. Huge growth area that's fully contained. And very simple compared to the Lonza facility, which is on the right, and a very dramatic difference in the cost of building the facility. What happens as you move into production is a huge reduction in cost of goods. And this is where it becomes very important to healthcare systems like in Brazil, Canada as well. But if we look at the blue part of this graph, we're looking at the cost of goods. So on the left is the production cost of a name brand drug that's produced in the US. Usually about 30 to 40% of the retail price that's shown on top in US dollars. If you look on the right, you can see our cost is about 10%. And so we can make a large profit for investors, or we can reduce our cost of drug in the market, outcompete other biosimilar drugs, and provide a benefit to the healthcare system, the provider, the government. So if we're looking at these plant-based systems, just to wrap up, I mean, why would you be interested in a plant-based system? Well, they're scalable. It's easy to build these facilities. The technology is known. The technology is transferable. We have transferred our technology from 0.5 grams of tobacco biomass to 300 kilograms production at a facility in the States. The cost of goods is significantly less, 10% compared to mammalian cell manufacturing, and 12 to 25% compared to bacterial. And the production time to get to having a process in place is considerably reduced. So we see a great opportunity for this. And in our program with Hugo and the Biomangenus, we're very excited about the possibility of moving this project forward and bringing a product to market that's going to provide a huge cost saving to the Brazilian and Canadian governments, and also a big opportunity for us to market that drug internationally. Yeah.
Thank you very much, Dom. Very good presentation. And we can see the many benefits that this platform can bring to the manufacturing of monoclonal antibodies and other biotherapeutics. Now we open to discuss. So if the people here in the auditorium want to to do questions, comments, please ask the microphone, or you can type in the chat. And people from connected virtually can also do questions and comments by the chat. Is there anybody that would like to initiate some question? Can do in Portuguese. We have translation, or English. Yeah, Procopio.
Hi, congratulations, very nice presentation. We work with monoclonal antibodies, and we have a doubt in this moment, what is the status, the situation about monoclonal antibodies glycosylation when they are produced by plants. We have some concerns, but some years ago, I don't know now, how is the situation about it?
That's actually a very good question. It's the key question to using plants. So plants have different glycosylation patterns to mammalian cells. So plants are eukaryotic organs like mammalian cells, but plants have specific sugars, xylose and fucose, which are specific to plants, and they're lacking galactose and another fucose, 1,6-fucose. So what we've done as a company is we have engineered out using knockdown mechanism the plant-specific glycosylation, and we've engineered back in the sugars from the mammalian system that are lacking. And we've also working with 2 different Canadian universities on sialation, where now most monoclonal antibody drugs are not sialated, but some are. And it's interesting to look at that for perhaps a biobetter drug. So, you know, there are 2 concerns. One is safety, and we feel we've addressed that by having mammalian type glycosylation. And then the introduction of the sugars, then we have to get confirmation that the actual patterns do provide the same binding characteristics to the targets where they're relevant to activity. And so that is an activity that we do continuously in our development program. We actually have 3 patent families covering this area. It's so important. Yeah. Thank you.
Thank you, Don. I have a question to Dr. Thomas. I don't know, sorry if I lost in your lecture, but what do you think is the most important criteria to select a CRO or CDMO in your view?
There is no straightforward answer to this because it depends pretty much on the specific needs of your needs or the company's needs. It's at the end which drives the decision is a mix of several criteria. But as the interaction of both the CRO and the company is essential for the success, if already at the negotiation, when you're negotiating with the CRO and discussing the technical capabilities and all of this with them, the communication is not good, then I would not advance. Therefore, as a first criteria, how open are they? How communicative are they? Is it easy with them to discuss? Do they understand my needs? If this already doesn't work, then I would not even go into the next round.
Good, thank you, Thomas. I have here a question from the people who are participating virtually. I believe it's a question to Don and also to me, maybe. Rodrigo asked, how far are we to produce biosimilars in Brazil using plant systems?
Yeah, I mean, we're very excited about the opportunity here. I think perhaps I'll pass this off to you, but I mean, we have been working in Brazil since 2014 on one biosimilar drug, and have now moved on to this candidate that's more relevant. And there's a need for introduction of technology that companies like ourselves and others can partner. And then there's a need for the development capability that I think Biomangina brings, and the facilities, which is a huge, exciting opportunity. But where it is, I'll leave to you.
Yeah, as Don said, we have a cooperation with Plant Farm. It's a strategy of Biomangina is to have alternative expression systems, and platform is one of them. And we believe in the benefits that this platform can bring. And also we are already co-developing biosimilars in this platform. And we believe in the next years, you're going to have this product with marketing authorization, and then pave this path to others biosimilars and other innovation products. I'm sure. Our time is over. I have one more question, please. Marco, there is another microphone to him.
I have a question about the seed. The idea produce the seed, or have some company supply the seed, modified seed, because these plants is a modified, have a glycosylation path humanized. Né? The idea buy the seed, or plant form or Biomangina produce the seed.
Yes, that's an interesting question as well. The answer is complex, so we need to have a bottle of wine and discuss it. But simply, Plant Farm is developing the seeds. We use two types of systems. We have a wild type that's generally available, and we've engineered actually a transgenic plant that has modified glycosylation. So some of the aspects of the modified glycosylation. So in this case, we could use a seed that's already present here in Brazil, or we will have to import the modified seed. And that's a question that we have to address with the regulatory authorities here, because that's what we'd like to do. Yeah.
So again, I will congratulations the lecturers for Dom and Thomas. Thank you very much to share your experience and work. Unfortunately, we have to close the session to initiate another session that will be the oral presentation for development and optimization of a protocol for 19BBZ CAR T cells generation, presented by Leonardo Ribeiro Batista Silva from INCA. Thank you too much.
Bye.